Drugs. Cyclophosphamide is an outdated chemotherapy drug once used to treat small cell lung cancer. It was displaced in favor of platinum-based drugs in the 1980s. Both kinds of drugs work at first but falter after a few months as the cancer develops resistance. Platinum-based drugs became the standard of care mainly because they cause lesser side effects, but they have not substantially improved prognosis. Today, the typical patient survives less than a year and a half after diagnosis. In this study, however, researchers showed that small cell lung cancer cells resist cyclophosphamide by activating a specific repair process, and demonstrated that throwing a wrench into the repair process makes the drug much more effective, at least in mice. The findings, available online in Cancer Discovery, suggest a pathway to better therapies for one of the least treatable forms of cancer. The problem is that these tumors respond to treatment initially, but then they come back. This has not changed for 30 years. These tumors are just massively resistant to just about everything. So what this study shows is that we can actually combine a new target with an old drug to reduce resistance and potentially make the treatment much better and give these patients a much better chance. The study came together fortuitously. A few years ago, his team discovered that a protein called RNF113A may play a role in small cell lung cancer, but the researchers could not determine what the protein does. The paper noted that RNF113A plays a role in the process. The team discovered that RNF113A is regulated by a protein called SMYD3 that is highly expressed in small cell lung cancer and some other cancers. High levels of SMYD3 are associated with more invasive disease, increased resistance to alkylating chemotherapy and worse prognosis. One set of cells came from a patient who had not yet been treated, so the cells had not had a chance to develop resistance. The other came from a patient who had been treated with and become resistant to standard platinum-based therapy. However, the combination of the two drugs stopped the tumors in their tracks. They did not restart growing for the duration of the experiment. We're talking to a number of other groups about starting a phase 1 clinical trial as soon as possible, Mosamaparis said. One of the challenges we will face is convincing doctors to go back to an old drug. But the nice thing about this strategy is that it may work where current therapies have failed. This treatment worked just as well against the tumor from the patient who had already relapsed on platinum-based therapy as it did against the untreated patient. Original written by Tamara Bhandari. Note: content may be edited for style and length. Cancer Discovery, 2022. OF1 doi, 10. 1158 2159-8290. CD 210205. Cite this page. Washington University School of Medicine. Drug combo therapy in mice blocks drug resistance, halts tumor growth, may lead to better treatments for deadliest form of lung cancer. Science Daily. Drug combo therapy in mice blocks drug resistance, halts tumor growth, may lead to better treatments for deadliest form of lung cancer. Science Daily. Thanks for watching. Please subscribe my channel for more videos.